Everyone, thank you for the invitation. I really appreciate it. It's a little strange to be talking in front of a computer. It feels like I'm talking to myself. It's very narcissistic. But anyway, I'll try to get over this. I'm trying to move. Do you know how I can remove the arrow? I'm trying to remove, oh yeah, this maybe. I have your figures on the screen and I couldn't see this. The idea of this presentation today is to talk about the link between ecophysiology, so the properties of organisms at the scales that are relevant to them. So the link with those phenomenon and biogeochemistry. And the example I'll take to illustrate this presentation is diazotrophic cyanobacterium. So the main topic of this presentation is point 3 here, meaning the link between physiology and biogeochemistry. But I'll start with a few words very briefly on phytoplankton first, and then on nitrogen fixation. I wasn't sure what your background was and whether you had more or less all the same lectures and classes in the past. So I'll just briefly introduce what it is that I'm going to use as examples. So we're dealing with phytoplankton here, and a very illustrative figure is the image, the satellite image that you have right now in front of your eyes. It's a very famous image that we see in many different books, and it's usually used to emphasize the importance of phytoplankton at the global scale and the fact that they contribute to half the primary production on Earth, etc. I'm going to use this image for another purpose. What you can see here is, okay, phytoplankton in the sea. First remark, it's not homogeneously distributed. So at the very large scale, we can see that there is a lot of phytoplankton in the sea, but it doesn't always appear at the same place and not in the same abundance. See how it's far more concentrated in the high and low latitudes compared to the oligotrophic gyres. So that means that at the very large scale, the development of phytoplankton is highly constrained by a number of environmental factors that change in space and that also change in time. If we get a little closer to this plankton, let's go to a schematic representation of the vertical axis of the water column. So you've got the surface here of the water and depth as you go down. What this diagram expresses is that all the environmental factors that the phytoplankton responds to are highly variable in time and in space, but also according to depth. So as an example, I drew the exponential decrease of light as we go deeper, and on the contrary, the exponential increase in the availability of nitrate and nitrite, for instance. If you look at temperature in the upper part of the column where there is mixing going on, the temperature is rather homogeneous and tends to be higher, and then it linearly, more or less linearly decreases with depth. So there are lots of factors that the phytoplankton responds to. So that means multiple limitations. And another important concept is the concomitance of those limitations, the fact that they occur together at the same time. And if we go even closer to this phytoplankton, now we're looking at one single cell, and I took the example of the unicellular diazotroph Crocosphaera watsoni. Here you've got a slice of Crocosphaera actually that's transmission electron microscope, so you can see the inside of the cell. And one cell is one entire organism, and it's 3 micrometer wide. So it's a tiny organism, and the scales, the time scales that are relevant to this organism are tiny. The first one is faster than the millisecond. The organism will react to the light that's and the photons that are hitting the surface of the cell. So within milliseconds, it can respond to this factor and generate ATP and reducing power, the energy blocks that will thereafter serve for all the downstream processes in the metabolism and in regulations. And those happen within minutes to hours. Then all those processes, metabolic processes, will lead to what we can express as the overall growth rate, and we'll observe whether there are divisions or not. So the division cycle is observed at the time scale of the day. And then on longer time scales, those cumulated growth processes at the daily scale will lead to population dynamics at even longer time scales, so weeks, months, even centuries. So there is this cascade of scales in the environmental forcing conditions of the phytoplankton that lead to emergent phenomena. And so when you observe a bloom at the surface of the ocean, what you observe is something that emerges, but it emerges when you look at it, you see instantly, okay, right now there is a bloom, but the conditions that led to this bloom, you have, if you want to consider those conditions, you have to go back days, and you have to go back all the way to the very instantaneous phenomena that might have happened weeks before. And all those cascades lead to what you're observing at one time point. So the consequence of this is that when you're looking at seasonal dynamics, what you see is something emergent, and it emerges from the coupling of longer timescales with elementary processes. By elementary processes, I mean growth processes, photosynthesis, respiration, for instance. And so a legitimate question then is, okay, but if we are to understand the appearance or the dynamics of a bloom, what scale should we look at, and what level of detail will matter at when observing phenomena at larger scales? This is an open question, and it's all the more relevant for those who work in ecosystem models, for instance. When you want to build an ecosystem model, what it is that you should represent in your model, what processes are important. So to illustrate the following presentation, I'm going to use phenomena that were measured in diazotrophic cyanobacteria. So I'm going to just say a few words. I know you've most probably heard a lot of it, but I'm going to say a few words just about nitrogen fixation. In the open ocean, the most abundant diazotrophs are cyanobacteria, and I'm mostly interested actually in diazotrophs that contribute to primary production. So I will not talk about all the heterotrophic strains, for instance. So when focusing on cyanobacteria that are the players among the phytoplankton that can perform nitrogen fixation, the one that's been known longest is a filamentous strain. It's called Trichodesmium. It's between 5 and 10 micron in diameter. And so it lives in filaments that can associate in puffs and stuffs. Beside this one, a little more recently, were discovered the unicellular diazotrophic cyanobacteria, and called UCN. Two groups were described one of them being represented by Crocosphaera. They are smaller than Trichodesmium. So because every living organism on Earth follow, well, there is sort of an allometric relationship between the size of an organism and how abundant it is on Earth. UCN are smaller than Trichodesmium, so we could think that they would most probably be even more abundant. And a third group was also thereafter described, called UCNA. This one is a little more mysterious. It's smaller than the UCNB and C, so potentially it is even more abundant. The first two are cultured today, and the third one is still not, unless I'm late on the literature, but I don't think it's been cultured yet. It's actually symbiotic, so it's very difficult to keep it in culture. So one conclusion that can be drawn from this very simple diagram is that we have here three groups, mostly three groups of players that have different shapes, sizes, abundances. They are physiologically different, so one could logically wonder if they are different, then do they lead to different fluxes of carbon and nitrogen? So if I'm back to a generic representation of a diazotroph, they all use light. So light can be a driving factor of their growth. They need nutrients, macro and micro. Since they can perform nitrogen fixation, they can also supplement their nitrogen requirements using N2. They're sensitive to temperature, more or less, depending on the strains. And they all have loss terms that can be internal, like respiration or exudation, or that can be external forces driving their death, like mortality, predation, grazing. I'm not going to dwell on the mathematical representation that this leads to. It's just a rate of change of the biomass in time expressed as the gross growth rate multiplied by the different limiting factors that one wants to consider, and minus all the loss terms. But this is not the purpose today. A very special feature of nitrogen fixation is that, okay, take nitrogen fixation, the nitrogenase activity, it allows nitrogen storage in the cell. Photosynthesis, on the other hand, allows for fixation of carbon and can lead to carbohydrate reserves. And the organism needs both. But another consequence of photosynthesis is the evolution of oxygen. And this one is seriously damaging the nitrogenase activity. It actually irreversibly destroys the nitrogenase. So the consequence of this is that in theory, there shouldn't be photosynthesis and nitrogen fixation at the same time in diazotroph. That gets very complicated for them to handle both. So in UCN, in the unicellular, they solved the issue by performing photosynthesis in the light, of course. And so they reserved the dark time for nitrogenase activity. So they separate those two activities in time so that the nitrogenase is not really exposed to all the oxygen that's been evolved during photosynthesis. And they respire a lot to get rid of this extra oxygen. So it's something that they have to handle, and it can lead to extra energy expenditure, in particular to get rid of all this oxygen. If we compare here a filamentous strain Trichodesmium and a unicellular one, you have the illustration here on this graph of what I just said before. So this graph represents represents 1 day, about 24 hours on the x-axis, and the y-axis is the ethylene production. It's just a proxy for the activity of the nitrogenase enzyme. The day is partitioned into equal lengths of light and dark. And for trichodesmium, the brown curve, the nitrogenase activity occurs mostly during the light. It falls down to 0 at night. And then you can see that in the following light period, it increases again. So in filamentous strains like trichodesmium, well, nitrogen fixation happens during the light. In the unicellular ones, here you have the example of cyanotheke, it's the exact opposite. There is no nitrogen fixation in the light phase, then it kicks in during the dark, and then it decreases again and falls to 0 in the following light phase. So there is a major difference between those two, even though they are both diazotrophic cyanobacteria. Trichodesmium can directly benefit from photosynthesis for the provision of ATP and reducing power. So that's a very efficient way to feed the nitrogenase enzyme, which is quite greedy in terms of energy and reducing power. On the contrary, in unicellular strains, since the processes are separated in time, the organism actually relies on the storage of carbon reserves during the day so that they can respire those reserves at night and produce the ATP and reducing power that the nitrogenase enzyme requires. So those two different physiologies necessarily mean that the organism will have distinct carbon and nitrogen fluxes, in particular related to the nitrogen fixation activity. So if they have different physiologies with different carbon and nitrogen fluxes, does it mean different consequences in terms of biogeochemistry at the large scale? Well, probably yes. So one conclusion to this is that if we are to pick a model organism of diazotroph, where it's a difficult choice because there isn't a single model organism that could actually act as a representative of all the diazotrophs. They're all different actually. So there is no unique or ideal model organism. So in the following part, to illustrate my presentation, I'll be using unicellular diazotroph, and the little name of this organism is Crocosphaera. So what we did is that we ran cultures in the laboratory. So we had duplicate cultures. We could not have triplicates because the vessels were so large. It's 5 liters in there. So we had at least 2 cultures, if not 3. And those cultures were successively exposed to 4 different light regimes. So these were 4 different experiments. In the first one, the reference, there is a balanced light and dark phases, 12 and 12. In the second experiment, we had a very long light phase, 16 hours, and a short dark phase. In the third experiment, it was the opposite, short light phase, long dark phase. And in the last one, it was a mixed light regime with fluctuating irradiance. So in the following graphs, the light phase is always the gray background and the dark phase is always the blue background. So what I'm going to show you here is a monitoring in the reference culture of the carbon cell quota, which is the carbon content expressed per cell, as well as the nitrogen cell quota, so the nitrogen content per cell, and the resultant molar C/N ratio. So looking at carbon dynamics, you have 2 curves because we had 2 cultures. So we can clearly see that there is a carbon buildup during the light phase, and then a decrease of the carbon content at night, which is logical regarding the fact that the organism needs to burn and respire carbon reserves to perform nitrogen fixation. Looking at the nitrogen cell quota, it's the reverse. Nitrogen is increasing at night. Of course, cells are fixing nitrogen. They fix at night, so the nitrogen content increases during the night and thereafter decreases during the day because this nitrogen is used to make up cell material, but it's also in part lost from the cells. As a consequence, you can see that there is a C/N ratio that highly fluctuates throughout the day. It goes from 4.5 to 10. So it's a very, very wide fluctuation. So there is a fluctuating stoichiometry in the cells. If we take the average value integrated from that curve, we actually end up pretty close to the Redfield ratio. Looking at carbon reserves that cells build up, so only the carbohydrate granules, those that are highly variable in cells, that show a very transient dynamics because they are actually it's like the tank, it's like a fuel tank that cells will fill whenever they can and burn whenever they need energy. So those carbohydrate reserves tend to increase during the light phases. Of course, they are simply replenished as photosynthesis goes on. And you can see how they dramatically decrease at night because they serve as energy source for the cell. So they are burned, respired, and consumed for the cell to perform the different metabolic processes and also to build up cellular material. A very interesting and striking feature in this organism is that in this organism, 30% of the total organic carbon was actually carbohydrates. It's like if a third of your own weight was just reserves. It's just enormous. Looking at how much carbon cells spit in their environment is also very intriguing. It is actually very significant. These are TEP, transparent exopolymers. So these are sort of sticky particles that are carbon rich that are found in the medium and that actually originate from carbon molecules that the cells exude and release from their body. One can see that there is again a diel fluctuation in the TEP in the environment, and we could calculate that about 10% of the total carbon in the cells is actually released per day, and this is quite significant as well. And it means that the cell is actually running fast, uptaking a lot of carbon, storing a lot of it, but in the process, it also loses quite a bit of carbon that is released in the environment every day. I'm just going to briefly evoke what we got in the other two experiments, so the long light phase one and the very short light phase one. What you can see here is again the carbon content, the nitrogen content, and the resultant molar C/N ratio. What's interesting to see here is that the average level of carbon content is higher here compared to the 12-12 regime, the reference, but it's even higher in the 8 to 16. It's a little bit counterintuitive because here they have a shorter light and yet they store more carbon. They also store more nitrogen. So it is even more counterintuitive because cells have less energy available because of the shorter night. But what actually happens in this experiment is that cells didn't like this regime. They didn't like it because they couldn't grow as fast as they would like or as they could. As a result, they were dividing much slower. And because they were dividing much slower, they kept building up cellular material from a light phase to the next, but they postponed the division cycles. So what happened is that they actually grew fatter because a much longer time occurred in between two divisions. So looking at what happens is at very short time scales. Here we're at the time scale of hours, allows us to really understand why the division cycle at the daily scale is actually shorter here compared to here, but why in consequence cells have become bigger. In another experiment, we pushed a little further and simply ran a set of batch cultures exposed to a light gradient to better characterize the light response of the organism. So what you can see on this curve is the irradiance that was applied to the culture on the x-axis, and on the y-axis, the growth rate that was observed in all those cultures. There is a set of subsaturating irradiances up to 300. And then what we can see is that the growth sort of saturates. Cells just cannot run any faster, even if we provided them with stronger irradiances. In the meantime, we quantified carbon and nitrogen content in those cells. So here are the subsaturating irradiances. And what you can see is that the nitrogen content as well as the carbon content keep increasing with irradiance. So the more light they get, the bigger cells tend to be, and the higher the carbon and nitrogen content. But also what we noted is that the gross carbon and nitrogen incorporation was, the gross fixations were higher than the incorporation rate. So not only the cell content increases, but the exudation rate increases as well. And at the highest irradiances that we tested, we realized that carbohydrates were not 30, but now 50% of the total organic carbon. So this organism has a storage capacity, which is just astounding. And in parallel to this, cells spat more and more carbon into their environment. So the exudation rate increased with irradiance. But an even more surprising feature is that this carbon exudation was constitutive of growth. Here we are in subsaturating irradiances. None of those irradiances saturated the growth rate, and yet cells kept spitting carbon, even though we provided them with less and less irradiance. And that's also something very counterintuitive, because usually carbon exudation is seen as sort of an outlet for too much carbon fixed. Here, we cannot have too much carbon fixed because cells are subsaturated. They could fix more, they could store more, they could grow faster, but they don't, and still they spit carbon. So this is a very, very interesting point of their physiology. And last, so this carbon exudation that happens at all irradiances, well, eventually turns into TEP in the environment. And so the TEP in the higher irradiances tested reached almost a third of the total organic carbon. And so that's an interesting thing to think of, because usually the bigger cells are considered to be significant providers of carbon for their environment. It's very well known that diatoms, for instance, exude a lot of carbon. Well, it's very interesting to see that even small cells like those diazotrophs that are supposed to burn a lot of carbon for nitrogen fixation, nevertheless manage to exude carbon in their environment, and that this exudation rate is really not negligible. So the corollary of all this, when we go to larger scales, is that the diazotroph exudes releases ammonium when fixing nitrogen. This directly stimulates the phytoplankton community, but bacteria also very much like ammonium. So the activity of diazotrophs actually also stimulates the microbial loop. Those bacteria, cyanobacteria, also exude DON, dissolved organic nitrogen, which directly favors the heterotrophs, but can also favor the phytoplankton community because more and more eukaryotic algae are discovered capacities for mixotrophy and also prokaryotic as well. So the exudation of organic nitrogen actually stimulates both communities. Last, we discovered that there is significant exudation of carbon, which stimulates the microbial loop and participates to export to a lesser extent. So that's the biogeochemical consequences of growing diazotrophically. Oh yeah, and I forgot the photoheterotrophy, sorry. So in summary, what I wanted to emphasize today is that in any organism, carbon and nitrogen metabolism will be tightly constrained at scales that are relevant for the organism. And those scales are tiny, very short, very quick. And there is then in diazotrophs and at least in the unicellular strains, this temporal segregation between carbon and nitrogen acquisition will necessitate efficient storage abilities. So that means that the organism must be able to fix much more carbon than it actually requires for its growth. And there is a possibly non-red field stoichiometry observed in those organisms when we grow them in light regimes that are not balanced. And because those organisms must acquire a lot of carbon, what happens is that they still exude significant amount of carbon, and they also exude significant amount of nitrogen. And this can be, as I said, counterintuitive, but in the end, that's actually the strategy that they took: fix as much as I can, store whatever I can, and what I can't, well, I'm going to spit it out. But they do this at all irradiances, so it's really constitutive of their growth even when they are subsaturated. And so in the end, what I wanted to illustrate today is that you can see how the ecophysiology, so small-scale processes in the growth of an organism, will actually drive the biogeochemical fluxes at the scale of the entire population. And I think I will stop here. I'm not sure what time it is. I don't